Life in the lab as a hardy contaminant. When Professor Gertrude Buehring steps into her lab in Koshland Hall, she is never met by HeLa cells. In fact, she makes a point of it. We never grow them, she says. I wouldn't want to take that risk, actually. Buehring, a professor of virology in the School of Public Health, has a reason to be wary of HeLa. Both her PhD and postdoc careers were spent working at UC Berkeley's cell culture laboratory housed in the Naval Biosciences Laboratory in Oakland, a cell repository funded by the federal government that characterized and maintained cell lines for research scientists. She happened to be working there at a time when Dr. Walter Nelson Rees, the co-director, was working hard to expose HeLa's misdeeds, the vigorous cells crime, the contamination of other less hardy cell lines. Nelson Rees was not the first to suspect contamination by HeLa cells. In the 1960s, Dr. Stanley Gartler, a research geneticist, released the initial HeLa bomb. Gartler had discovered that the 18 different cell lines he had collected for his research all turned out to be genetically identical, with genes only present in people of African descent. HeLa was a suspect, but many scientists refused to accept the implications of his discovery and chose instead to ignore it. 10 years later, Nelson Rees picked up where Gartler left off and discovered several HeLa-specific chromosomal markers that could be used to test the identity of cell lines. Since this repository had so many cell lines, Nelson Rees was going through all of them and examining them for these markers, recalls Buehring. While he was there, he came up with 40, which was more than one expected. In that instant, any tissue-specific research that used the cell lines identified as HeLa contaminants were suddenly invalid. How can research on breast cancer cells be taken seriously when the cells used were actually cervical cancer cells all along? It has been estimated that over 500 research papers and more than $20 million of funding have been wasted. The problem stems from the adolescent days of cell culture. After Dr. Gay established the HeLa line, everybody was so excited and thought they could establish a human cell line too, Buehring explains. Unfortunately, most of these labs did not have the knowledge or equipment to properly culture cells. What they did have was plenty of HeLa cells around, and due to HeLa's hardiness, a single cell could outgrow and overtake all normal human cells in a culture. Suddenly, everybody was able to establish a human cell line, jokes Buehring. Turned out that many of them were just HeLa. Even years after being exposed for what they really were, HeLa contaminants continued to be sold by the American Type Culture Collection, ATCC, one of the largest international cell line repositories, and scientists continue to request the cells they've become so familiar with. In fact, many scientists were hostile toward Nelson Rees and unable to accept the implications of his work. Over time, however, the ATCC refused to sell HeLa contaminants. This doesn't mean, however, that they are no longer used in labs today. Since her days working at the Naval Science Laboratory, Buehring kept HeLa in the back of her mind and was curious about the extent to which HeLa contaminants were still used, as well as how aware researchers were about HeLa's potency. I couldn't find any research papers where people actually looked at that, she said. So in 2004, she decided to look into it herself. With the help of an undergraduate student, Professor Buehring conducted a survey of researchers known to culture cells and asked what kinds of cell lines they worked with, whether it was for tissue-specific work or not, and if they ever tested the identities of their cell lines. The results surprised her. There were so many people who used HeLa cells in their laboratory, and only about 50% did any kind of check to see if there was contamination, she says. Not only that, but about 60% of respondents had acquired at least one cell line from another laboratory rather than from a repository like ATCC. Oftentimes, people will think they're getting a good cell line from a colleague down the hall, but they don't know it's already been contaminated, she explains. If it isn't checked, you never know that. Buehring herself rarely gets cells from other labs, but if she does, she makes sure to check their identity before trusting them. The survey also revealed that about 10% of respondents still used HeLa contaminants, 30% of which used them for tissue-specific purposes. The original HeLa bomb of the 1960s and 70s had lingering effects, it appeared. The truth is, however, that many researchers today don't see HeLa as a contamination threat anymore. Back when cell culture started, they were using glass. It was so easy to contaminate, says Fisher. The use of disposables today helps eliminate some of the threat. Nowadays, I don't worry about that at all. Like Buehring, Fisher also insists that getting cells from reputable sources like the ATCC. Otherwise, she suggests verifying their identity. Going back to frozen stocks of cells every week or two is another method of avoiding contamination. Zhang says he, too, is not concerned. If it gets contaminated with a different cell line, it's very recognizable because looking under the microscope, every cell line has a very distinct shape, he explains. HeLa cells, for instance, are often very large and triangular. Not only have cell culturing methods improved, but HeLa's days as the easiest and fastest growing cell line are over. New cell lines have emerged that work just as well, if not better, for certain applications. Insect cells, for instance, can also be used to overexpress proteins, but can be grown in larger quantities. This makes them ideal little protein factories for when researchers need large quantities of a given protein for study. People don't use HeLa cells as much because they're harder to grow than insect cells, says Fisher. Believe it or not, harder to grow. Bacteria cells are actually the easiest cell type to grow, but may not be capable of making some of the more complicated human proteins that often require more intricate modifications before they become fully functional. Yeast cells, which have a more advanced protein assembly system, are the next line of attack, followed by insect cells. Only if these three cell types are unable to produce the human protein of interest do researchers consider human cells such as HeLa. There are other reasons that HeLa cells are finding themselves at the bottom of the list. As a cancer cell, its DNA is a major liability. HeLa cells have the strangest karyotype, says Fisher. They have 3 copies of this, and 2 copies of this, and 5 copies of that. They're not normal. Many researchers today choose to work with cells that more closely resemble normal human cells, thus taking their in vitro systems one step closer to mimicking how a real human functions. IMR90 cells are one such example. Cultivated from the lungs of a human fetus in the Netherlands, IMR90s have a normal karyotype with 46 chromosomes. Of course, there are drawbacks to working with mortal cells. They only go up to 60 populations and then they die, says Fisher. We have to thaw those every 3 weeks. Not only that, but as the cell line becomes older and older, they show signs of aging and may not be ideal for research anymore. Luckily, Zhang doesn't have to worry about trying to work with more normal but finicky cell lines. Since we're looking at such a fundamental process in the cell, we think that although HeLa cells are very different from normal human cells, the basic processes that keep the cell alive should essentially be unaltered, reasons Zhang. Buring agrees that there is an important distinction between using HeLa to obtain basic cellular material versus using HeLa as a whole cell and expecting all of its cellular processes to be the same. As a bag to hold the biomolecules of study, however, they work just fine. For other purposes, HeLa may not be top dog anymore. A wealth of information, making research faster and easier. Like a celebrity, the more scientists learn and work with HeLa, the more popular it becomes. It began in the early days of cell culture when HeLa's vigor and human origin made it unique. Today, scientists take comfort in HeLa because of its familiarity. It's well characterized because so many labs have been working on it, explains Zhang. There are many tools that work with HeLa cells that don't necessarily work well with obscure cell lines. This is because many of these tools or techniques were originally developed using HeLa cells and are thus optimized for them. One example, gene knockdowns, can be used to stop HeLa from expressing a specific protein, thus helping Zhang determine if and how it affects the secretory pathway. HeLa also has good transfection efficiency, ensuring that when Zhang transfects his HeLa cells with a protein, a higher percentage of the population will have his protein of interest. Not only can HeLa do a lot, we also know a lot about it. As a human cell line, the Human Genome Database becomes an important source of genetic information. Zhang, for instance, uses the database to design his knockdowns and target a specific gene of interest. Though not crucial, HeLa's specific genome would make things even easier. Luckily, this is now a possibility. Since August of 2013, researchers can submit proposals to gain access to the HeLa genome on the National Institute of Health database. You would know which genes are expressed instead of empirically testing it, which can take a week, says Zhang. Access to the NIH database would let him see what genes are expressed and to what degree, therefore making it easier to design effective knockdowns. Simply put, HeLa cells are just plain simple. A wealth of information, crossing the boundaries of privacy. As HeLa's popularity in the lab grew and the list of medical discoveries reached the ears of non-scientists, public interest sprouted. Articles began to surface that speculated on the identity of this mystery woman. Credit was given to Helen Lane or Helen Larson until eventually Henrietta's true identity was revealed. Researchers, members of the media, and con artists soon hounded the family, all hungry to use them for their genetic information, family history, or as pawns for fraudulent money-making lawsuit against Johns Hopkins, respectively. No one, however, provided the family with any information in return, and they were often left in the dark about their mother's final months, the origin of HeLa, and the implications of HeLa research. The infamous cells became a burden. This kind of disclosure about a human cell line would be unthinkable today, and rightly so. Nowadays, if you take their cells, you wouldn't call them by that patient's name because of confidentiality, says Fisher. What if you found a genetic abnormality that could be traced back to the family? Which is, in fact, a very real question posed by members of the Lax family. In March of 2013, HeLa's complete genome was published without the family's knowledge. Researchers like Zhang sit on both sides of the fence on the issue. It would be helpful to get some genetic information that would be specific to HeLa cells, he begins, but there's this privacy issue, an ethical issue. The genome was removed after the family voiced its concerns. A few short months later, however, a compromise was reached known as the HeLa Genome Data Use Agreement. Researchers can obtain controlled access to the genome after submitting a proposal, and any data obtained from the genome must be openly shared on the NIH database. Access to the genome will be tightly regulated by a committee of 6, 2 of whom are members of the Lax family. It may not have been a simple journey, but the family is in the dark no more. Over the years, the family has come to learn about the use and importance of HeLa cells, and the research and medical communities have in turn learned to respect them. Credit in large part must be given to Rebecca Skloot, whose book, The Immortal Life of Henrietta Lacks, was the first to focus on the story of Henrietta and her family rather than HeLa. Skloot took a different, more constructive approach than the scientists and members of the media who came before her. She chose to work with the family rather than use them, and helped them understand all parts of their mother, from life to death to HeLa. In turn, scientists can now learn about the history of the cells they've become so familiar with in the lab and understand their significance outside the lab. The wealth of information that we've learned from her cells is just so overwhelming, says Zhang. Not only has this information resulted in valuable medical advances, research papers, and PhD theses, but also in crucial laws and policies governing the use of cells and tissues and a greater awareness of cell line contamination. It may be that with good comes bad, but the key is converting the mistakes of the past into something constructive for the future. Zhang would argue that acknowledging those who deserve the credit is also important. I think we should all be grateful to her.